because I didn't even know you were doing. I mean, I think yeah, you had the issue with the dude that you were supposed to talk to, but I didn't know that you were actually going to be doing another hangout, so I just started one myself. And then I'm like, dang, Chu has a hangout going, and then I've had one going, so
Well, I was expecting the Rose to come, but he didn't show. So for my viewers that are
yeah,
listening now,
I one myself.
well, he didn't.
And I'm like,
Oh,
dang, Chu has a hangout going, and then I've had one going.
I got some feedback.
So what were you and Arcane talking about?
Okay. Well, what we're talking about is chromosome 2 fusion and intelligent design. I decided to start this hangout because the Rumpus was supposed to show up, and he didn't show up. So I guess what my stake on this would be is that I disagree that the chromosome 2 fusion, the site where they're claiming a telomere-telomere fusion, it didn't happen. And if that were to happen, that would actually be very dangerous for the organism. It would trigger a phenomena that we call apoptosis, so it would cause the cell to self-destruct. So, I mean, like when you look at the information that's involving, you know, most of these fusion events, there's a lot of different chromosomal fusion events, but usually they result in satellite DNA with satellite DNA, or satellite DNA with telomere DNA. There's no known cases of, well, every known case where there's a telomere-telomere fusion, it's very dangerous. It's a very dangerous thing to happen. I think that they've found some fusion events like that in yeast, but that triggers what's called tumor igenesis, and that's obviously, this is something that's involved with cancers. So whenever this happens, it usually triggers apoptosis, and when apoptosis happens, that causes the cell to self-destruct. It's another mechanism, like I believe the P57 gene, where the P57 gene, it's programmed to, whenever there's a malfunction, like a cancerous cell, it'll cause it to self-destruct. It's kind of like that. It's a mechanism built inside the cell to keep unhealthy cells from reproducing.
Wait, does that mean that chromosome 2 fusion will be a fusion of unhealthy, what you just said?
Well, no, there's no known case of a telomere-telomere fusion like that, like what's being claimed in humans. There's no known case like that. There are fusion events that happen, but usually whenever there's a fusion event like that happens, it's satellite DNA to satellite DNA, or satellite DNA to telomere DNA, but there's no telomere to telomere fusion. So inside of the cell, or inside of the 23 claimed chromosomes that we have, they're trying to claim that will be one of our chromosomes, chromosome 2 fused. That would be the first ever documented phenomena. And when you actually look at the fusion site, there's actually a very important transcription factor, or sequence, a specific DNA factor that controls the rate of information to DNA to RNA. So the site that they're actually referring to is actually it's actually a very important part of the chromosome. It's just one big chromosome, but they're picking out one part that is actually the start of a very strong and important mechanism.
Oh, okay.
Right. So chromosome 2, the two chromosomes, they didn't fuse like they were saying. It's part of the chromosome already. But if that happened the way that they're claiming, to an end-to-end telomere fusion, that would trigger more than likely apoptosis, and it would self-destruct. That's dangerous for the body or for life. So when you really listen to what they're talking about, there's about 64,000 genes around that area to where the binding site supposedly occurred. Most of those binding sites, or I don't want to say binding sites, but those sequences that you're finding in that location, they are actually referred to as pseudogenes. A lot of these sequences, we don't really know what they code for. Those sequences, they're not found in any other apes on the planet. So I mean, they got a lot to work here before they can prove that it's actually a binding site. There's nothing indicating that it is a binding site or it is a fusion event like they're claiming. Everything is indicating that if it happened the way that they're claiming it did, like these two chromosomes fuse, it would trigger an apoptosis event. But I think Arcane, he had a pretty good video that I actually shared the other day. I was actually speaking about this with Hawkeye the other day, and Tony was in there, I believe. But during the time I was actually talking with him, I was doing my homework, so I think, you know, there was a lot of mixed-up confusion. This very actual video is what I showed the other day, and I think it's actually a pretty good video. So we're going to try to screen share that. Tell me if you guys can see when I'm screen sharing. All right, can you guys see it?
I can't see it yet.
Okay. And again, for everybody's listening, the link's in the description and it's in the side chat, so you can all come on in and talk about what you'd like to talk about. Can you guys see it?
Yep.
All right.
Can you hear me?
Yeah, I hear you great, man.
All right. Yeah, I'm just going to play where he starts talking, the geneticist. Can you hear it?
Apes have.
Will you try to screen share it? Because I've actually got it up right now.
Yeah, y'all both got the same video up.
Yeah, he just shared the link right here. I'll just share it, Arcane, since I've got it up, right?
24 pairs of chromosomes, but humans only have 23 pairs. But it is assumed that humans descended from the great apes. It was claimed then that two chromosomes fused together into one at some time in the past as humans were evolving. It was believed that human chromosome number 2 was the resultant chromosome composed of 2 fused ape chromosomes.
And I want you guys to pay attention to this, too. This guy is a PhD in genetics, so he knows what he's talking about. This isn't just like, you know, some average and nobody. He's actually very educated on this field.
We discussed this claim previously on several shows, as well as Crevo Rant number 181, and how the chromosomal fusion argument is bust. More news has come out since the airing of these shows and our previous interview with Dr. Jeffrey Tompkins, who joins me again. Dr. Tompkins' PhD is in genetics with a master's in plant science and a bachelor's in agriculture education, and he was on faculty in the Department of Genetics and Biochemistry at Clemson University for a decade. He also ran his own Genomics Institute for 5 years. He is now a research scientist with the Institute for Creation Research, where he continues to publish various papers. Some of those papers we're going to discuss today. He joins me from his office in Dallas. Welcome to the show again, Dr. Tompkins.
Thank you. Good to be back.
Let's start off. I want to backtrack to a paper you came out with in 2011 addressing this claim that humans have a fused chromosome, which allegedly supports evolutionary claims.
Right.
So let's go back in time for a minute, back to 2006, and revisit a lecture given by evolutionist Dr. Kenneth Miller at Case Western University. The lecture is entitled The Collapse of Intelligent Design. Now, it's interesting to note he very confidently claims that not only is there evidence that humans had 2 chromosomes fused, but that this is such powerful proof of evolution that he brought it up in the Kitzmiller-Dover trial. A court case which frankly wasn't a court case, but a 3-ring circus complete with a circus judge. A circuit judge. The segment of Miller's lecture where he discusses the chromosome fusion was linked to all over the internet. In fact, even today, I get comments from atheists and skeptics pointing me to that segment posted on YouTube under the provocative title, How to Shut Up Pesky Creationists. They hand me this video as if I haven't seen it before, as if it's absolute proof of evolution. I'd like to play a segment of that lecture so we can hear straight from the horse's mouth exactly what Miller said.
What must have happened is that one pair of chromosomes must have gotten fused. So we should be able to look at our genome and discover that one of our chromosomes resulted from the fusion of two primate chromosomes. So we should be able to look around our genome. And you know what? If we don't find it, evolution is wrong. We don't share a common ancestor.
Okay, so in 2011, you and Dr. Jerry Birdman came out with a paper showing that this alleged fusion site was small, highly degenerate, and really not much of a fusion signature.
Right.
2 years later, you published a second paper showing that the fusion site actually contained the genetic code for an important feature inside a highly expressed life-supporting gene. Now, we discussed this when you were on the show last, and I got a firestorm of emails and comments saying that you were wrong, and even Kevin Miller said you were wrong. So clearly you were wrong.
Okay, well, should we backtrack and
Sure.
and cover the details of that discovery? Okay. Well, the fusion site actually is a functional feature inside an RNA helicase long non-coding RNA gene. So this gene produces functional long non-coding RNAs of various sizes. Now this gene has 3 exons, or 3 coding regions. The other areas of the messenger RNAs are spliced out of the transcripts made from this gene. So you get short variants and you get long variants of this gene. So Ken Miller actually gave an interview to a reporter that works for, what was that magazine? I've got it here, Mother Jones.
Oh, yes.
Magazine. Does that sound familiar?
Yes,
okay.
it does, yeah.
So the reporter was a guy named Mooney. So Mooney basically got the interview from Ken Miller. So Ken Miller says that the fusion site is actually 1,300 bases away from the gene.
Right.
So a friend of mine at the Discovery Institute said, Jeff, did you see this article? And I said, no, I didn't. He said, well, this is what Miller is saying. He said, you need to contact him and get this thing settled. He said, you know, Ken Miller never responds to me whenever I send him an email, but maybe he'll respond to you. So I sent Ken Miller an email, and he actually responded within an hour, very quick. And he was very cordial. He gave a nice long email explaining his position. But the interesting thing was he obviously didn't read my paper, because if he would have read my paper in its entirety, he wouldn't have said the things that he said. So I basically had to explain my entire paper to him over a course of several emails. And then he finally realized that he had made a mistake and that I was right. And so he said in the last email, talking about the long transcript, not the short transcript, the long messenger RNA produced from this gene that has all 3 exons. He says, in this transcript, the fusion site is in the middle of the first exon, as you know. Well, actually, it's not in the middle of the first exon. It's in the middle of the first intron. But anyways, so he acknowledged I was right. And then Casey Luskin actually took this piece from this email where Miller acknowledged I was right and published it in an article in The Blaze, which is an online media operation. And so people can actually get this article that Casey Luskin published with Ken Miller's quote from the email admitting I was right. So basically, everything is exactly as I described it, but not as I'm describing it really, but it's because that's what the data says. Yeah, one thing that's interesting about the fusion site, when they first discovered it back in 1992, now when I talk about when they first discovered the DNA evidence for it, when they sequenced what's called a DNA clone from human chromosome 2 from that region, when they found these telomere repeats, even though they were degenerate, it was not what they were looking for. They say that in the paper because they know that when fusions occur in nature in animals that actually survive these things and sometimes even reproduce afterwards, that these fusion events always involve something called satellite DNA. So you can have, actually the PowerPoint presentation I gave you has got a picture describing it. So you can have a chromosome break and have two regions of satellite DNA fused to each other. You can have a telomere fused to satellite DNA, but you never see telomeres fusing to each other in living animals. Sometimes you will get telomeres that will fuse to each other in cancer cell lines, which is, I think it's safe to say that's not a healthy
No, definitely not.
living tissue. But I have evolutionists always say this. Yeah, well, look at cancer cell lines. Telomeres fuse in there. Well, that's not a healthy living tissue indicative of a healthy organism. Cancer genomes are completely rearranged and completely haywire.
Right, definitely not onwards, upwards
Because-
evolution.
No, they're not. They don't represent,
Right.
functional normal cells. So anyways, we never see these things in living animals. So when they discovered this degenerate telomere region, it was not what they were looking for. They were looking for something that had satellite DNA in it. So if the fusion, if the human chromosome fusion was real, it would be the first documented instance of a telomere-telomere fusion ever occurring in nature.
Right.
But obviously that's not what it is.
Right, because two halves of the gene aren't going to come together from two chromosomes.
Well, telomeres are designed, actually telomeres are highly functional machines at the end of your chromosome.
Right, right.
They contain a very complex set of proteins that are grouped together called the shelterin protein complex that sits at the end of your chromosome, and the DNA at the end of your chromosome actually folds back into itself to form a DNA quadruplex, and then the shelterin complex sits down governing all of that and encasing all of that. And it's not just a static complex, it's actually a functional set of active proteins that also interact with elements of the cell cycle and various pathways inside your cell. So telomeres are actually functional machines, entities at the ends of your chromosomes that are doing all sorts of important things to maintain genome stability and to keep your cells functioning properly.
Right, right.
So they're not just these bare ends hanging out there waiting to fuse.
Right.
So they're designed not to fuse, basically.
Yeah.
When you think about linear chromosomes compared to the circular chromosomes in bacteria or archaea,
Right.
you have a logistical problem at the ends. You have an engineering problem that must be solved, and not really a problem, but
Right.
from our perspective of intelligent design. But if you were a human engineer, it would be a serious issue to have to deal with and to solve.
Right, okay. Let alone blind chance.
Right.
So pesky creationists didn't shut up about this. All right. So if we can back up just for a second, because so the viewers can understand what we're talking about here. You said the fusion site produces a non-encoding RNA.
Well, this gene does not produce a protein. So there's over twice as many genes in the human genome as there are protein coding genes that produce RNAs. And these RNAs do all sorts of things. Some of them stay in the nucleus. Some of them go out into the cytoplasm. A very small percentage are even used as signaling molecules in what are called exosomes outside the cell. But a lot of these long non-coding RNAs function in the nucleus, and they modify DNA. They're called chromatin modifying RNAs. And then a lot of them also combine with proteins to form these big complexes with proteins. The RNAs sort of act as backbones for these proteins to bind onto, and they also contain regions that bind to DNA as well in the genome. In fact, there was a paper a few years back where this particular secular research paper said that the genome is like an RNA machine. So really, it's an RNA world in the nucleus of a cell, in the genome. And so this gene produces long, non-coding RNAs of various different types. It's alternatively spliced. It has dual promoters. It has a promoter in front of the gene. Promoters are like switches in genes that turn the genes off and on and are involved in causing these genes to produce transcripts or RNAs at certain levels and certain amounts at certain rates in the right tissues at the right time. And so it turns out that the second promoter in this particular gene, it's called the DDX11L2 gene. The second promoter in this gene is the fusion site. So this is not just DNA sitting there doing nothing. The fusion site, and I have to use the term fusion site because it's really not a fusion site. It's a promoter inside a gene. And it's very common for genes to have promoters in their first intron inside them, as well as promoters in front of the gene. This is common across the human genome. And another important thing to understand is that you have genes on both strands of the DNA molecule. DNA is double-stranded. You have a plus strand and a minus strand. When we annotate genomes, we always annotate genes as to whether they're on the plus strand or the minus strand.
Oh, darn it.
So the great ape.
Telomeres are not our
Well, I'm sorry, guys. I accidentally clicked out of that. But I think that's enough anyways. I think he pretty much made the case. The video seemed to be almost over. So what do you guys got to say on that?
I think it's another example of evolutionists jumping the gun and proposing things that fit the theory that, you know, a couple years later we're discovering things that question it. So, you know.
All I gotta say is that I gotta get more into this because I don't really know much about chromosome 2 fusion.
Yeah, I agree. Same here. That's why I joined this hangout, just to learn more stuff. And yeah.
So what's your take on this, Hawks? I believe you were in the room with me the other day. We were talking about the same exact issue.
Sorry, that was an agonizing delay. I think everyone's done that. I was eating some soup, and then you can't find the right window. Where's my unmute thing? I only heard. I think people have seen. I just popped in during the last few seconds of it. Now I had heard that video before. I think in the hangout that True was mentioning. I'd mentioned this Tompkins. I'd forgotten his name. And I said that I'd seen this interview with Ian Juby, and then True Empiricism was showing that clip, and that's actually the same conversation. So I'd seen it before a few months ago, and then True Empiricism had played it through on this hangout the other night on True Talk TV. So even though I didn't see much of it now, I should be familiar enough with it to sort of, you know, figure out what's going on. My understanding of what Tompkins' concern is, and I would agree if this were the case, this would be a real problem for this idea that it's a fusion. That is, people are saying that human chromosome 2 is actually a fusion, an end-to-end fusion of two different chromosomes that are typical amongst apes. Is that you would expect to see in this long chromosome 2, in the middle of it, you'd expect to see a telomere and a telomere, and those two bits of telomere would be kind of like leftover telomeric sequences and wouldn't be there to code for something. I have to correct something I said when we were talking about this the other night. I said, according to Tompkins, there's actually a protein coding gene sequence that goes right through those telomeres, and that would be like, that would be pretty bad for this whole idea that it's an end-to-end fusion. And I've since been told and confirmed that I'm incorrect about that. It's not a protein coding gene that goes through it, but rather a promoter gene that goes through it. But it doesn't matter. It's the same problem, right? I just had the wrong bit of information to mind. But the point is that if you've got, like, your telomeres are like the end caps to a chromosome. And if you take two chromosomes and you stick them end to end, you'd expect to see, well, here's a chromosome, and then these two end caps kind of stuck together, and then some more chromosome. And you would not expect there to be any type of a gene, whether it's protein coding or promoter or controlling, it doesn't matter. You wouldn't expect there to be any gene that goes through these telomeric sequences. So the fact that Tompkins says, well, yes, he's discovered in reviewing the sequence that there is actually this gene that does pass through, that's a pretty serious blow for the idea that this is an end-to-end fusion. But as I say, he's the only one that holds that position. Everybody else who's looked at it says no, the gene he's talking about, which they always refer to by letters and numbers, DD2 something something, is actually beside the telomere, and that they're saying he's misreading the genome. That gene is there, but not passing through the telomeric sequences.
So is he lying when he says that Kenneth Miller sent him an email and agreed that are these gene sites at the so-called fusion site?
No, I don't know if he's lying about that. He's lying when he says that Ken Miller admitted he was wrong, and that was a very odd thing to say, I thought. That was earlier in the video when he says, oh, so I emailed Ken Miller, and Ken Miller wrote back, and it was clear he hadn't read my paper, so I had to explain it. And then he says he had this email exchange with Ken Miller, and where Ken Miller admitted he was wrong. I found that whole thing to be completely uninformative and lacking any credibility, because he wasn't saying, well, I was discussing this with Ken Miller, and at the end of this conversation, we agreed that this promoter does something, 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 and actually went and told us what the subject of it was. He was just so gleeful to say, and I've got an email from Ken Miller where Ken Miller says that he was wrong and I'm right. Well, wrong about what? Right about what?
Well, he actually described what he said he was wrong about.
He said, oh, go ahead, sorry.
Yeah, you can play it back. I was listening quite carefully because I was thinking maybe, he would produce the email so that we could have that proven. But basically, he did describe the site or whatever they were discussing. I'm not sure the technical jargon.
The transcription factor binding site. That's what it'd be. I think.
Yeah,
I think specifically what they were criticizing is there's about 64,000 sequences that are found in that location, and those 64,000 sequences, they're not found in any other apes. So if there's nothing similar with that, if you would to look at it from the 23 chromosomes, chromosome 2 fusing, you would be able to tell the sequences, but those sequences were not found in any other apes. And more specifically, when he was talking about the telomere-telomere fusion, he was trying to point out that does not happen anywhere in nature, and whenever it does happen in nature, it triggers apoptosis. Because usually when something like that happens, you're dealing with tumorigenesis, and that's dangerous for cells. You find that in cancerous cells. Usually whenever you're looking at a fusion event, because they happen all the time, it's either satellite DNA to satellite DNA or satellite DNA to telomere, but you never find a telomere-to-telomere fusion specifically because of that reason, that it would trigger apoptosis. So I think that's specifically what he was trying to get to. What they were looking for, if it were to be a fusion site, would be a satellite-to-satellite DNA fusion, or a satellite DNA to a telomere fusion, but that's not what they found. They found a telomere-to-telomere fusion where you're finding a transcription factor sequence that's, where you're dealing with a specific DNA factor where they control the rate of information to DNA to RNA. That's what you're finding there. You're finding a specific part that's actually very, very important for the chromosome. So I think that's what's important there. The patterns and sequences that you're looking at throughout the before and after the chromosome, they don't fit with any other chromosomes that you would expect to find in apes. If there was an end-to-end fusion, the pattern isn't there.
So all that stuff that you just said, was that Like, do you recall? Because what we were just talking about was this idea that Ken Miller had sent an email to Tompkins where he said, "Tompkins, you're right and I am wrong." That's Tompkins' specific claim. And I'm saying I don't recall what Tompkins was saying that Tompkins was right about, and Ken Miller admitted he was right about it, and that Ken Miller admitted that Ken Miller was wrong about it. So you just said several things about how end-to-end fusion doesn't happen, or if it happens, it leads to all these problems. You talked about apoptosis. Was any of that stuff part of what Ken Miller allegedly conceded to, saying, "Oh, I'm wrong, and you're right," or was that a totally different?
Well, I'm not
time-stamped on the side.
Yeah, that's Arcane's. That's Arcane's criticism. My criticism is more so what he was saying about the information. I think that's all that's important is that fact that if you're expecting to see a satellite-to-satellite DNA interaction, that would actually fail with the prediction if that's what they were looking for. They didn't find. And if all we're ever seeing is satellite-to-satellite fusions or satellite-to-telomere fusions, we don't ever see a telomere-to-telomere fusion, well, then we can't ever say that, I mean, like that's something expected. What you find in humans is not found anywhere except for, I believe, yeast and other, cancerous, cells, to where, and I'm not talking about yeast being cells or anything like that. I know this is the basic unit of life. I don't want anybody to jump on that little point that I said right there. But all I'm saying is that that would have triggered a tumor type of scenario inside the cells to where they would become cancerous, right? So that's what I'm trying to get at. This little scenario to where you're seeing a telomere fusion, it's not expected. But if you were to see something like that, you would expect that to be dangerous for life.
I mean, I don't know what to say. We talked about this. I've addressed all of those points, and I addressed all of those points in an on-air hangout that was polite and cordial and open, and both sides got to put their points out, and has since been deleted, making that whole conversation pointless.
No,
and now you want me to come in again and address those same points again, so that what? If I go through it again, then and you don't like the way how you appear or look in this conversation, you'll delete this hangout too? Why would I participate in that sort of a form of dialogue?
Okay, first of all, I don't know why you're getting mad all of a sudden, Hogtie.
I'm not getting mad. No, stop. If you're going to say dishonest things, I'm not getting mad. I'm making a point. I'm punctuating my point.
I know, Hogtie. Hogtie, the video wasn't deleted, actually. I already told you that I was going to separate it into two videos, and I just actually went through the process of putting the whole entire end at the beginning. There was 3 sections that I wanted put out separately so that I can span out the content on my channel. And during the time when you were speaking with me, I was actually doing my homework, so there was a lot of things that were not touched on that are being touched on right here. Do you remember when I went off, when I had to take the video off a screen share, I had to pull up my homework documents. I actually told you this over Skype. So there was a lot that we didn't touch on. We were just talking about specific sequence patterns, and we didn't get into any detail over that. I just allowed the video to play so that I could do my homework while the video was playing, and he can make his case for you. And I think it would be a little more fair if you just deal with what I'm saying right here, because this is.
Well, I just say, why do I want to do it again? We just had this whole conversation.
No, we didn't have any conversation over this. We didn't talk about it. We didn't talk about tumorigenesis, and we didn't talk about what happened with the telomere-telomere fusion, what would happen if with satellite DNA or satellite-to-telomere DNA. None of this we were actually talking about.
Yeah, we had the whole conversation where you said what you would expect. You were saying you would expect 2 functioning centromeres, and I said, well, I don't think that would be the case. And
well, obviously, we didn't talk about that in technical detail. I mean, we just mentioned that point like towards the end of the hangout. This was like 3 in the morning, My behavior towards that hangout was like, when a kid's coming to you and they're bothering you for a candy bar and not really paying attention to what they're saying. That's what it was like for me. I was sitting back doing my homework and I wasn't really paying attention too much.
Okay, so it's your view that.
So, I mean, if we just mention just that you would expect to find one centriole here, and we won't talk about it. Obviously, we need to talk about that, right? Yeah. It's like everything else we haven't. Where I mentioned just now that I wish you would actually respond to that we didn't talk about in the other hangout.
No, because for a second, I do want to address that point specifically, because you don't seem to recognize what you've done. You'd sent me a message on Skype, which I always welcome, except in the last while I've sent you messages on Skype. You haven't replied. I've invited you to Hangouts. You haven't replied. You sent me a message saying, hey, if you want to talk about this, and you sent me a Hangouts link. So I joined only to discover that it's an on-air Hangout, and that now we're fired up and we're going to have a debate. And apparently it's a repeat debate of something we had the other night. By your characterization of it, you could have done better than you had done in the other conversation, so you want to do it now. At no point since that discussion up to now, including right now, have you said, hey, are you busy? Do you have a minute? Hey, Hogtie, you want to do a Hangout?
Stop the drama for a second. Stop with the drama.
There's no drama. I'm busy.
This Hangout doesn't have anything to do with you. Hold on a second. Hogtie, stop, please. This Hangout doesn't have anything to do with you. This Hangout has to do with because of the fact that myself and Lumpus were supposed to have a discussion right now, and he didn't show up. So I saw your message that you sent over the YouTube, and I figured, hey, he was in the discussion the other day. Maybe he would like to come in so we can have a chat. That's why I get this now. Yeah. But I mean, I kind of feel like you're being a little unfair right now. I mean, you can at least deal with the points that I'm making, because we didn't talk about any of this stuff the other day. All we talked about was the fusion event. I talked about the patterns, and I alluded to that just now earlier when I talked about the 64,000 sequences, and we shared, the video. That's all we really did. There's a lot more substance that I got to right now that I wish you would actually talk about.
Well, there's more than an hour of that video deleted. Again, I'm getting a wicked echo.
It's not deleted. I don't know why you keep on saying it's deleted.
Okay. Because the video was shortened using the online video editor. That's why I said that.
Yes, it was. You were absolutely right. It was shortened, but downloaded and cut into sections.
Okay, so you did download it first?
You guys jumped the gun after I did that, and I do this all the time. People can see where I take sections out, and I make sure that I upload them on my channel. I section them out. But the only reason why I did that was because I wanted to span out content on my channel. It was a really good discussion, and we had 3 different topics, and I wanted to make sure I can cut up the video so I could have three pieces of content on True Talk, because there's nothing there.
Okay,
but who cares? You're bringing in drama.
Okay, I accept that's possible. Let me recognize then. You referred to this. What did you say? I was angry or whatever. I mean, obviously, I'm certainly annoyed.
Let me respond. You're just bringing in drama for a second.
No, I'm not. I'm actually about to dial it back. I'm trying to apologize. Okay, don't let me apologize. Whatever. Go ahead.
Hold on. I just want to respond to what he said. Emmanuel said that what is satellite DNA? I think that's actually really important. Usually you find satellite DNA in chromosomes 1, 9, and 16. And with satellite DNA, that's basically where you're having a sequence or base sequences repeat throughout the genome. That's what you expect. And I think that I think actually a little bit over half of the satellite DNA in mammals is satellite DNA, and I think that actually a lot. Correct me if I'm wrong. If anybody knows about that, I think that a lot of it is actually junk DNA as well.
Okay.
So I just wanted to respond to that, because if nobody knows what satellite DNA is, they need to know what it is anyways. But I mean, Hogtie, I just, that was like unexpected.
Okay, well, I can dial it down, and I can explain just ever so briefly. Sir, can you put that over here? Just do it over here, because otherwise the noise goes to the table. Yeah, I'm sure I sound a little bit prickly about it. I will acknowledge I'm having a bit of a bad day. I have a bunch of work that has to be done before I do another road trip coming up here, so I'm actually really, really busy today, and I apologize that I come in and sort of be a little bit bitter about it. My impression, and if you correct me on this, that it just looked to me like you removed that portion of the video and then told people that you were going to put it up separately. If you say you did in fact download it before using the editor to remove it off the main video, then that's fine. I accept that, and I apologize for and actually my recent post where I said that I deemed it likely you were lying about that. That's wrong of me to say that sort of thing, and I apologize for that. But I guess can we just for a second? I don't want to turn your whole hangout into this. Arcane, kiss my ass. I saw what you typed in the side chat. What the drama is related to is we tend to have, and I want to let you know ahead of time, True, that I'm not trying to turn your hangout into this. I just want to give the context here, and that is you host these hangouts where you guys sit around for 4 hours and go, oh, well, we put out the link and nobody's coming in. We put out the link and nobody's coming in. They must be cowards. And whereas I send you invitations ahead of time through Skype. I let you know hours ahead of time, hey, I'm going to have a hangout. I'm thinking of this topic. Hey, what do you think about this topic? Do you want to do a hangout on that topic? You've never invited me to a hangout. You've never said, hey, I think Hogtie's interested in this. I think we should do a hangout on this topic. What I get is an hour into a hangout, you'll say, well, nobody's coming in. You're all cowards.
I'm getting a little irritated now. I mean, I gave you a bunch of points right here about the satellite DNA and the telomeres and so on, things that we did not talk about in the other discussion. You've got my full attention now, so maybe we should talk about the actual substance.
I have one single point to say
I
in
don't want to go
response.
into drama.
Okay, one single point to say in response. I am working right now. I'm at my computer because I'm working. You'll notice that I've put up maybe 2 posts on Google Plus all day today. I did that one because I saw a response pop up on my notification thing. But I'm sitting here with my drafting software open. I'm like doing actual work. And again, I'm going to apologize because I think I'm going to go back and listen to this part of the hangout and see, am I that guy who when I'm at work I get all grumpy and I get rude with other people? Because I don't want to be that guy. So I apologize for that. But I mean, literally, I'm at work. I accepted your message because I considered, well, I thought you wanted to talk about something. I didn't realize you were inviting me into a debate. So I'm going to go. I'm going to thank you for inviting me, and we can talk about this another time. And I welcome you to, after I leave the room, for you and Arcane to sit around and talk about what a drama lover I am and what a coward I am because I wouldn't talk about this stuff. I'm happy to talk about it any other time.
Well, I got to say, Hogtie, but this is very strange. I mean, you came in pretty respectful, then as soon as I started talking about it, you went on to drama. I mean, like you just averted trying to get into.
Yeah, because I'm annoyed, and I've apologized for this. I was annoyed that you deleted the video, and you've explained, and I've apologized for it. And again, I'll apologize. That was wrong of me to say that.
Okay, well, just drama. I came in here because I wanted to talk about this actual topic. Anyways, drama. No more drama at all. Anybody who would like to talk about this subject matter, if you guys disagree with what I was saying, come on in and let them have
Yeah, and normally I would, if I mean, I'd be doing this for at least, and it's only 4 o'clock now, at least a half hour. But and if I do come back in, I will come back in and apologize. Okay. I get on well with Arcane, and I don't like it getting into this sort of a tone, but it's food for thought. Don't sit here and call people cowards because they can't come running to your hangout when you haven't even invited to it. I think that's fair, don't you?
This is an open hangout, Hogtie. You left a message just previous to this, and because of that, I gave you the link. And I told you why I gave you the link, because we were talking about this type of subject matter.
Yeah. So. No, so what I'm talking about the others, but whatever. I'll let you guys get on with your. I don't want to get into the drama, so I'll let you get on with your other stuff. Okay. Oh, good. Tommy's here to talk about what a coward I am. All right, we'll see you guys.
All right.
Bye, Hogtie.
Thanks for coming, man.
That was very unexpected. It wasn't expected. Yeah. So what's up? What do you guys got to say about it, huh? You guys think that chromosome 2 fusion, that's a chromosome 2 fusion event or evidence for evolution? What do you guys think?
I think it'd be a miracle of evolution.
I say the evidence for evolution is weak.
MMR, what's happening, man?
G'day, mate.
But like I said, I think it more than anything points out how quick they are to jump the gun and say that this is evidence for evolution, and then a couple years later, it's debunked with new discoveries. It sort of seems like they have, more than anything, a confirmation bias when they're doing their so-called science. It's like, we know that evolution happened, so let's go out and find evidence for evolution.
And then when it's against,
yeah.
And then when the evidence is against it, they will deny it without providing any evidence as to why it's wrong.
I just joined. Have you guys brought up the Tompkins thing where he believes that this said fusion event happened within a functional gene?
Yeah, we just played the video of him.
Okay, sorry. I haven't seen any of it.
Yeah.
Okay. What did Hogtie have to say about that?
But nothing. Yeah, I think that he was expecting, because in the last discussion I didn't really get into any technical details.
Mm-hmm.
because at the time I was multitasking. So in here, I don't think he was expecting us to bring up the subject matter, in a little bit more detail. So I think that just came off to me as sort of being defensive. I don't know. But.
Have you heard anybody in the evolutionary? I've got an echo of somebody from the
sorry,
evolution.
that's me.
That's okay. The evolution crowd actually confront Tompkins on this concept that this is happening within a functional gene.
Yeah.
Because he's about as high profile a creationist / ideist out there, and so I would think they would be eager to pounce all over that. If Tompkins made a mistake, someone in that community, thousands of them that there are, would be all over it like flies on a rib roast. But I haven't heard of any such thing.
Well, first and foremost, Kenneth Miller, he was on that video saying basically that Kenneth Miller retracted his initial claim and that he agrees that it is a functional gene site or whatever at the so-called fusion site. At least that's what I got from it, and that's why he put it out there. So if there was ever anyone to challenge it, why not Kenneth Miller, since he's clearly going public and saying Kenneth Miller was wrong? The only thing I've heard, though, is that he got it wrong, that this gene site is actually like a long ways away from the so-called fusion site. So I've never seen any papers yet.
Now, hold on, boys. Can somebody help me understand the implications of this whole fusion thing?
Well, they believe that, humans, apes have 48 chromosomes, and we have 26 or 46 chromosomes. We have 23 chromosomes, 23 pairs of chromosomes, right? So they're saying that one of the chromosomes fuse, chromosome 2 fuse, and that's evidence of evolution, because that's what they would expect, but that's not what they would expect. They would expect a satellite DNA to satellite DNA fusion, or satellite DNA to a telomere fusion. No one would expect a telomere to telomere fusion, because a telomere to telomere fusion triggers an apoptosis to where it causes the cell to self-destruct. It's dangerous. You find that in cancerous cells. So the fact that you're seeing a telomere, the fact that they would say it would be a telomere to telomere fusion, it's not true. I mean, there's nothing indicating that this was a fusion event for evolution. On top of that, the 64,000 sequences that you find before and after this supposed fusion event are not found in any apes whatsoever. They're not found in any apes. So there's a problem with it all over the place. The centrioles, I believe centromeres, they don't find two of them. I mean, there's so many problems with this idea. So the point is that this isn't evidence for evolution. You don't find any of the same sequences that you would find in any of the other apes that have the 48 chromosomes. If this was a telomere to telomere fusion, but we would expect that it would be dangerous for life. It wouldn't be happening like we're seeing here. So they're not. What you see there is not what you would expect if two chromosomes fuse together like they're claiming. You would expect a satellite to satellite DNA fusion, a satellite to telomere fusion, but you would not expect a telomere to telomere fusion. That's not found in any organism, and whenever it is found, it's dangerous for life. That's not to say that you don't find fusion.
You keep saying that.
That's.
You keep saying that, but it's not the case. It's not the case. It's worth noting, I have to chuckle, that I was out of the room for about 45 seconds before you accused me of lying. I thought that was great. Literally at work, I've now closed that software.
I'm more interested in what you said beforehand, where you said that's not the case.
Okay, but I just want to say, it was funny that even when I said, look, the whole point here is I'm at work. That's why I was a little short with you. I apologize for that.
All right.
As soon as I leave, you told people.
I don't want to take all time. I'm going to have to start muting because I don't want this to turn into drama. You keep coming in here and making it drama.
No, let's go to the telomere thing then. Let's go to the telomere thing then. But it's fair to point out when you do something like that. That's ridiculous. But okay. So what you just said was that you don't see telomere fusions. Yes, you do. There are other examples of it in nature. It's incredibly rare, but they're there. And you said that we would expect to see two centromeres. What do you base that on?
Can you give me an example then? Can you give me an example of another telomere fusion that does not trigger apoptosis or the situation to where you would see tumorigenesis?
Okay, so in addition to the examples I gave you the other night, there's the one in yeast that we talked about. There's one in yeast that we talked about the other day. I had 6 examples that I posted in the side chat during the last hangout.
Well, I mentioned yeast today, but you didn't mention any of those in the other hangout.
Yes, I did.
No, you didn't.
Yeah, I did. Yeah, I
Well,
did.
no,
I did.
you didn't.
I did. You were
Okay, well, give me the examples.
Can you
I just
just-
explained the problem with yeast. I just explained the problem with yeast already. So, I mean, do you have anything else?
Yeah, and then in any case, you keep saying that we don't see two chromosomes. We wouldn't see two chromosomes. I mean, the chromosome 2 would only function properly with precisely one centromere.
No. I'm saying that's not exactly what I'm saying at all. You see chromosome fusion a lot, but you do not see a telomere-to-telomere fusion. And when you do see telomere-to-telomere fusions like that, they're dangerous for the cell. And what happens when that ends? Unless what is it called?
How does it cause apoptosis though?
Telomerase. Unless that happens, and that that might. That actually helps with cancerous cells as well.
Can you give me a mechanism of action where it causes apoptosis within the cell?
Can I give you a mechanism? The mechanism itself is apoptosis.
No, how does the fusion or how does it cause apoptosis?
What does it matter how? I mean, if they recognize, hang on a second. If they recognize or see that it actually causes that, what does it matter how it happens?
No, it matters how it. He's right. It matters how it happens because whenever a malfunctioning cell happens, the mechanism is apoptosis. It's the same thing as the p57 gene as well, or is it the p53 gene? What happens is that when a cell indicates something that's not supposed to happen or there's something damaged, it'll cause the cell to self-destruct in a way. So
and how does the chromosome event?
The mechanism of how it happens is through apoptosis.
No. The apoptosis is the result of cell death, right? But how is that caused by the fusion?
Happens. Do you know what tumorigenesis?
How does it cause?
I'm trying to help you understand it. Do you know what tumorigenesis is?
Yes, but how is that following from the fusion event?
Because if you know what tumorigenesis is, you'll understand when telomerase breaks down why that's being triggered because of these telomeres.
Don't even attempt to
if tumorigenesis triggers apoptosis,
how does tumorigenesis?
Hey, stop it!
How does tumorigenesis come from this?
Stop it! I'm trying to answer you. You need to wait, okay? The telomeres, when they malfunction, whenever telomerase malfunctions or messes up, that is the reason why apoptosis is triggered, because of the telomeres. So you're asking me.
What's the function of telomeres? What's the function of telomeres?
It's like asking me. It's like.
What's the no, ask the question. What's the purpose of telomeres?
What does it matter?
It matters a lot for this. What we're talking about. What is the purpose of telomeres? How do they cause apoptosis when they fuse?
The telomeres, when they fuse together, that's not supposed to happen. That's the point. When the telomeres they fuse together, or when they touch with each other, like what's happening inside of cancerous cells, it malfunctions. They're supposed to be a sort of how do you put it? Almost a non-functional, like the parts, right?
I didn't answer the question. Ask what do telomeres do?
No, I'm answering your questions, but I don't think you understand
what do they do? You haven't answered that.
Telomerase, when telomerase happens because of the telomeres. When they're touching together, they malfunction, and that's what triggers.
But what's their purpose?
The question isn't this. You keep on asking me the same question. I tell you what happens during apoptosis.
You keep
on asking me about telomerase.
No,
stop. MM Artist, here's what the question is. MM Artist, stop it. Here's what the question is. The question is, MM Artist, here's what the question is. Look you're going to stop this right now. The question is that what is the evidence for this fusion event actually happening? Because that's not found anywhere in nature. The only time you see this happening, like in yeast, for example, that triggers apoptosis, because when the telomerase gets messed up and the telomeres are messed up, this is going to cause the cell to become more cancerous and to start to produce tumors. This is what happens when you screw around with the telomeres. That's what we know happens. The question is that what event do we know that can get around that issue, right? But we don't. That's the point. We don't see that anywhere. Anytime we see a telomere-to-telomere fusion like that, it triggers apoptosis. So what we would be looking for is a satellite-to-satellite fusion, or a satellite DNA to a telomere fusion. We would not be expecting a telomere-to-telomere fusion.
Okay, again, what is the purpose of telomeres?
Did you just seriously ask me that question again?
Yes,
It's the
because I want to know
structure
how
at the end of a
it causes
chromosome.
apoptosis.
It's a structure at the end of a chromosome. But that's not what we're talking about. We're talking about what happens with the telomere when it interacts with another telomere on this level.
One moment.
One moment, what? Did you just come in here to ask me stupid questions like that? Do you know what we're talking about?
How's a stupid question?
Let me ask you a question.
I'm responding to Emmanuel in the side
You know
chat.
what's? Hold on a second. What is a transcription
I
factor
don't.
sequence?
No.
What is the transcription factor sequence?
You're not flipping this on me. I want to know in which mechanism. How. It's important.
You're gonna answer my question. What is the transcription?
You still have not answered mine.
What is the transcription factor sequence that you find located right over there on the telomere?
You haven't answered the question of how it causes apoptosis.
It causes apoptosis because it screws up the telomerase, and because of that, it causes
mechanism.
Shut up. All right? It screws it up because it causes tumors to keep on reproducing. That's why. And in order for the cell to protect itself, it triggers apoptosis to self-destruct, causing cell death. So my question to you, no.
Dude,
I rest my case.
You need to quit jumping.
I got his volume decreased. I got his volume decreased. He's going to start answering some questions now. There's a transcription factor sequence-specific DNA factor that's located right where the supposed fusion event happened. Right? Why are we seeing that there, where the supposed fusion event happened? Explain yourself. I'm going to turn up the volume. You're going to answer this question. Go and answer the question.
I rest my case. You could not explain it.
Rest your case.
So,
You don't rest. No.
I rest my case.
What is your case?
So following that, I also hold on. Let me just.
Okay, so I got his volume decreased. Here's the problem right here. You have no case. You asked me pointless questions about telomeres, and you were given an answer. After I told you the reason why telomerase happens and why the tumors keep on reproducing, you kept on asking the question. The point isn't this. The point isn't your misunderstanding on apoptosis or your ignorance on apoptosis. The point is that why are we seeing a transcription factor region right over there located at the supposed location where this fusion event happened? Can you answer that question?
And you're being a hypocrite, sir.
Again, no, Antonio, shush, silence little one. Silence little one. So,
you shut up.
As I said before, I rest my case. And in the side chat, I provided you two papers. I provided you two papers titled "The Nature of Telomere Fusion."
You asked the question. No. Hold on, Antonio, just really quick. You asked the question, and I want you to directly answer the question. Okay? If you don't understand it, don't BS me.
Guys, let him talk, okay? Let him talk.
Thank you, Antonio. Thank you, whoever said that. So, I rest my case on that. Jesus Christ, Antonio, shut the fuck up.
No. Well, you need to be quiet already and answer the question, I'm artist.
I said I rest my case on that, and I took a screenshot that I provided two papers in the side chat, which I want you all to read.
You don't ever read your papers. No, shut up. You're going to answer the question. Stop it.
I don't need to answer your silly question.
But your volume's been decreased again. Answer the question. He says that he doesn't need to answer my question, and he's muting me now because he can't answer the question. Let's keep that in mind, guys.
The question, if you...
Okay, I kicked him, guys.
That was going to go nowhere.
Yeah.
Look, all I was saying is that he didn't want to be interrupted, but he can interrupt people all day. He was a freaking hypocrite.
Well, yeah, he just came in here to disrupt. You see, a lot of these people, they like to get carried away like that. Keep that in mind, because that same guy right there goes inside of a lot of these rooms with Steve and some of these other guys, and they run their mouth a lot. But that's what happens when they come into a room and they're actually being criticized with the BS that's coming out of their mouth. That's what happens right there.
I have a question about the fusion event, and maybe I'm missing something, but why, if assuming that that it did happen or it could have happened, why could it not have happened within the human lineage as opposed to in the common ancestor, which is what they're asserting, I suppose.
I'm sorry, I was reading the side check. Can you repeat your question?
Okay, so the fusion event, could it, assuming that it happened, just for the sake of argument, could that not have happened within the human lineage, say back in early man's history, for some reason? And so, if that's what indeed happened, how would that provide evidence for common descent?
No, I don't think it would happen. I don't even think it would happen in humans through just a naturalistic process. I don't think that would happen at all. I mean, because like I said, whenever you see telomere-to-telomere fusion like that, it's dangerous.
Yeah, that may be.
Yeah, so I mean,
that may be, but
look,
I think they found.
I'm saying if it did happen. Sorry, Tommy, I'm not sure which two things you're contrasting. What I think I understood with your second option was wouldn't it have happened in the human line?
Right.
I think that's actually what they're suggesting, is that it happened if you sort of map out, like make your little forking tree diagram of the apes.
Okay.
Gorillas and orangutans are sort of off to one side, and then chimps, bonobos, so that there's Pan, and then humans, and then the chimps and bonobos are off sort of on their own little branch that forks out. And it's only the human branch that had seen this chromosomal fusion, because all of the other apes have 24 pairs of chromosomes. So it would only be the human. Or alternatively, it might have been a slightly earlier ancestor than human, but something that went extinct. That is, I'm saying if there would have been a fusion before there's a thing that you would call human, and then it branched out, well, then the other branch is obviously gone, because we don't have any other creatures that would fit the definition of ape that also have the same number of chromosomes that we do. All the other apes have 24 pairs.
Mm-hmm.
Did I misunderstand your question?
No, I think that makes sense. Yeah. I can see what they're saying. I was thinking that they were saying it happened in the common ancestor, but no, that's not right.
Yeah. Well, I do want to address what true empiricism is saying about, well, it can't happen. I think he said repeatedly, it can't happen, because when there's a telomere-telomere fusion, it always leads to problems. Apoptosis or death, or I think the other day he was saying retardation was one of the options. And it's a strange kind of inferential way to approach science. If you say, look, we have lots of examples of weird things happening with chromosomes that tend to lead to problems. By and large, if you're a betting man and you see something wacky happen with the chromosomes, obviously I'm being very general, right? There's lots of different things that can happen with chromosomes. One missing, getting an extra one, non-disjunction, crossovers that are incomplete and one piece ends up breaking off. Generally these things are really, really not good. If somebody finds an example of where it wasn't deleterious or outright fatal, then you don't just say, well, this can't happen. The thing I'm looking at right in front of me can't happen.
So but he was asking for the science. If you had any actual science to show that to not be the case, that it is almost or always detrimental. So if that's what the actual science shows, and that's only what the science shows, then that's what we really should be going on, right?
Yeah, exactly. I would like to know a case to where this fusion event happens to where it's not dangerous for life. I mean, if you can give some good examples of that, then you can say that this type of fusion event would happen. But I think that there's also some other problems because of the sequences that you find before and after this fusion event. I mean, they're not found in any of the apes, and so nothing similar to 48 chromosomes versus our 46 chromosomes.
You've been given examples, and now you're showing up to say that you haven't been given examples. So I don't know what to do with that. Let's try this.
Well, you can give the examples because you didn't give me any examples as to what was correct. You didn't give me any examples at all.
I gave you 6 the other day, and if you want, I can take a moment and research them. And remember, I've said before, I'm not like a chromosome guy. I don't have a mental library of all these things. If you ask me for examples, I'm going to do what I did the other day and Google it. I mean, when I Googled it, I ended up finding 6 examples that were on the first page. So I pasted in some of those links. I can do that again if you think it helps. Let me make a different point then. Let's say that I can't find any examples. In fact, let's say that nobody could find examples because there just literally are none. And it turns out that like every time we've ever seen a telomere-telomere fusion, it's been something bad that's happened. But here we have a case where we're looking at human chromosome 2, and it's very clearly a telomere-telomere fusion, end-to-end fusion. Then we would say, well, that's surprising. I would not have expected that. Even if the human chromosome 2 fusion, as a result of the fusion, is the only example of a really destructive chromosome fusion, Well, then that's what it is. It would turn out to be the only example we know of. That's not the case.
I know.
No.
That's not.
Okay, why
first
is
of all,
that
it's not. You can't just declare it being a fusion event because
No. You can't.
You can't just say that we can look at this and say, stop.
You cannot, and I'm not.
Okay, well, that's the point here.
Don't put words in my mouth.
No. The point here is that it's not a fusion event. It's one chromosome. The point is that if it were to fuse from point to telomere fusion, like you're claiming, then what would happen? I mean, all of the examples that we know of, like in yeast or any other examples, it triggers an event to where it becomes dangerous for the cell or for any living organism.
Okay.
So that's the point here. We can't just say, well, this one happened.
Because
based on all the data available, if it were have happened that way, then it would have been dangerous. So it more rationally follows that it wasn't a fusion event until we have data available which could tell us that it actually was a fusion event.
Okay, we do. We do have data that tells us it's a fusion event. So, but let's say, okay, there's two. We can talk about cancer in a minute if you want to go back to that point. You've raised that a couple of times, but at this point, I'm trying a different tack because I don't know what to do when you say you ask for examples and then I give you examples, and then you say you haven't been given examples, and now you want me to go and search for more examples.
You haven't given me examples.
I have given you examples. I have given you examples. If you didn't click the links, I can't help you.
What's your best
example? What links? I mean, you keep on saying you gave me links, but you haven't given me any links. Not in the last segment or in this one.
Yes, I did.
Can you please give me the links right here, please? I mean,
why don't you go back and watch the last video? You'll see it. Yeah, even when I said there are several links, I told you here are 6. One of them is yeast.
Hold on. I'd like people to go to True Talk TV after this, and we can go watch that discussion. And none of this happened. There was no links given like this. I think the only things we brought up were the man in China that had that fusion event happen, and the yeast. But I think you brought up yeast. I don't remember actually specifically if you brought up yeast, but I brought it up here.
A minute ago you said I did.
You did not give me 6 examples of that.
I did. But okay, what I'm trying to do, like, again, this isn't.
If we're going to say, though, I just want everybody to keep in mind that's watching before we continue. If we're going to say that there are examples and we're not given any, well, then we have nowhere to go in that direction to actually say that it's true. That there are examples of this type of fusion event, a telomere-to-telomere fusion event, that is not cancerous or dangerous for life, that would not be triggered by apoptosis, has happened before. Okay.
Okay. Okay, we can talk about cancer because I do think that's an interesting. There's an interesting question in there. But what I'm trying to do, and I'm going to say, not successfully, it's not working at all, is I'm trying to say, let's approach this a little bit differently. You're asking for examples. Again, it's my position that I've provided examples. It's your position that I haven't. Whatever. Let's move past that. I'm saying, what if it were the case that I didn't have any examples, nobody else had examples? I'm saying, let's take a look at the logic of that sort of scientific inference. Let's say that you were a doctor or a surgeon. Your entire life, every time you saw a patient that was shot in the chest and the bullet hit his heart, that patient died. And you knew, just as a matter of course, a bullet going into someone's heart means that person dies. What happens if one day after 40 years of practice, somebody comes in and they're shot in the chest and the bullet went into his heart, but that patient's not dead? They're breathing, they're obviously in a great deal of distress, they need emergency surgery. Under your logic, you're going to say, well, gee, all the signs are here that this person's still alive, but he's been shot in the heart, and I know that that causes death. Therefore, this patient's dead, and you're going to zip him up in a body bag. That's the logic that you're pitching here. So
that's not the logic that I'm pitching here at all.
No, it is. And when I said this before, when I said,
hold on a second. There's examples. Hold on a second. There's examples of people getting stabbed in the head or in the heart and getting hurt in real dangerous spots, but they live, and usually afterwards they're having to have surgeries their whole life. Or
I'm not here to debate what the bullets do to the body. It was an illustration.
Here's the point with this. There is no example of any safe telomere-to-telomere fusion. There's no example of that whatsoever. All examples are dangerous. All examples show that they're dangerous, and they're dangerous for life.
Okay, and I'm saying, as a matter of logic, it doesn't matter that every example you're familiar with, I'm saying, even if I were to concede the point, which I'm not conceding, even if I were to say, okay, fine, every example you know of a telomere-telomere fusion is a big, big problem. I'm saying that, and let's say even scientists believe that when they did the sequencing and they looked at this thing and they said, hey, we've got a chromosome that's in our own bodies that turns out to be a result of telomere-telomere fusion. That's weird because every other time we've seen it, it's been deadly.
I think you're missing the point.
They would say, hey, that's interesting.
Yeah, I think you're missing the point. The chromosome
really
does
not.
not show a telomere-to-telomere fusion.
It sure
If
does.
the supposed site was a telomere-to-telomere fusion, what we're saying is that it would be dangerous, but the site is actually a very important part for something else. That's what we're trying to say.
Well, according to one person. And remember, I was the one who brought that up in the other hangout. Now, I know you were familiar with it beforehand of Tompkins' work, but you remember in that hangout, I brought up, I said, remember, and I asked you, I said, like, do you even remember the guy's name? You must be familiar with this. And I couldn't remember the name. You didn't know who I was talking about. But then you said, oh, actually, I know somebody who talks about this, and it turned out it was the same guy. So let's be clear. You and I are both familiar with Tompkins' comments about this. He says that the thing that let's call people pro-fusionists, if you want to say that. There are people who believe that chromosome 2 is a fusion. They will point to certain facts about the chromosome to come to that conclusion. And Tompkins says, well, no, it can't be a fusion because there's a coding region that goes right over through those telomeric sites. That doesn't make sense. He's the only one who says that. Everybody else who's looked at this claim says, no, that gene that's coded is coded beside this region.
You're saying that he's the only one who says that. When this first was discovered, everybody got superimposed on this finding, right? But new information showed that to actually be wrong. That's what the point is. So you're saying that it's only one person that's saying that.
Yeah.
And if it doesn't, let's just make this perfectly clear. If there's just one person making a statement that is a fact, it doesn't matter if every single person on the planet disagreed with that fact, it would still be a fact, right? So if one person is pointing this information out, it wouldn't matter. And I disagree with you that it's only one person, but if it was just one person saying this like you're claiming, then it wouldn't matter, now would it?
Yeah, well, it would, because other people have gone into have actually looked into it. I think what you're trying to do is to do this weird sort of global skepticism thing where you say, well, it might be the case that one person is right and everybody else is wrong. Like the first guy that, I don't know, whatever. Think of any scientific fact. The first guy that this occurred to, the first guy that thought of a benzene ring. Right? Nobody else thought of that. So
I disagree. I think that what you're doing. I think that what you're doing is.
I'm not doing anything because you're talking over me. So you can't tell me what I'm doing because I'm not doing it when you're talking.
Okay. Well, I think that what you're doing is actually an ad populum fallacy. I don't think
you're wrong about that. You're wrong about that. And I explained to you in the hangout the other night why that's not an ad populum fallacy. Because if what you're appealing to, the premises of the argument, appeals to the underlying evidence that those people use to form their opinion, then those are the premises. If, on the other hand, if your argument simply is, well, a lot of people believe this, and generally when a lot of people believe something it's true, then therefore it's true. That would be an ad populum fallacy. But that's not what I'm saying. I'm saying that there are a bunch of people in the world
who exactly what you said.
That's not exactly what I
just said just now that he is the only person who says this. Everybody else disagrees.
I said other people have looked into it. Other people have looked into it.
What you're doing,
there are other people in the world who know how this software works, right? Who know how? Have you looked at the? Have you gone to?
Hold on,
have you taken a look through the
hold on.
sequencing software and looked for the sequencing of genes?
Hold on. The difference between what we're saying, between what the two of us are saying, is that you're just saying a lot of people disagree. I'm trying to point out certain.
No, that's not what I said.
I'm trying to point out certain information, and I'm asking you for information that discredits what I'm saying. You haven't said anything other than, well, oh, he's the only guy that says it. More people have looked at.
That's not true. That's not true. I've said other stuff, and I've offered that we can talk about cancer. Let's then talk about, okay, why is it then that people are saying, or people have said, even before Tompkins, that when they look at this thing and they say, the thing being chromosome 2, they look at chromosome 2 and they say, huh, that's weird. That actually looks like two chromosomes that are stuck together. On what is that based? How do those people, why did people before Tompkins come to the conclusion? Like, it's a weird conclusion, isn't it? Like, you wouldn't just look at a chromosome and be...
Well, because the chromosome is larger than the other ones, obviously.
Joe asks,
what does it would look like double the size of the other ones because it's bigger, so they superimpose on that.
Trivia question. How do they number the chromosomes?
How do they number the chromosomes?
How do they number the chromosomes?
Why are you asking me this?
What makes 23 number 23? What makes number 11 number 11? What makes number 2 number 2?
I'm actually not 100% sure on that. Is there a point to this?
You're not 100% sure? Are you 4% sure? What's your answer? You don't know. Okay, you don't know. That's fine. They number them by size. Number 2.
Well, number 2 chromosome. Number 2 chromosome is bigger than the other one. So why would you? What is the point of this?
Number 2, you're gonna love this. Number 2 is the second biggest. Nobody suggested that chromosome 1 is a product of fusion. The reason is when you look at chromosome 1, you see telomeres, and again, I'm just going to restate in case new people in the audience join. When we keep saying telomere, we're talking about a sequence of non-coding, just an end cap. It's like the DNA doesn't just sort of dangle out loosely in the ends. The telomeres are like sequences of DNA. Sorry, that echo, thank you. They're like the end caps. And chromosome 1 has a telomere, and then a whole bunch, it's a really long chromosome, a whole bunch of stuff, and then it has a centromere, and then more stuff, and then a telomere at the other end. Chromosome 2 has telomere, some stuff, a centromere, some stuff, and then telomere, and then some stuff, and a vestigial centromere, and then more stuff and a telomere. And it looks exactly like it's got that same pattern, telomeres on the ends, except the two ends are stuck together. That was the first clue that it was actually a fusion.
No, it doesn't. No, that's actually not true at all. The 64,000, the 64,000 sequences that you find before that and after that, they were not expecting that because they were actually dubbed pseudogenes, right? So that's not what they were looking at all. When they were looking at the chromosomes, they didn't really know what they were there for to begin with. They didn't match any other apes that we find on the planet. So if there were going to be a chromosome-to-chromosome fusion like you're claiming, then we would be able to see that. We would be able to tell with the sequences that we're following.
No, even by this
they're not found in any other ape on the planet.
Even by this point, they could lay out the karyotypes. Even by this point, they could take the karyotype of, for example, chimpanzee. And for the audience's benefit, karyotype is not sequencing. It's when you look at the DNA when it's condensed into chromosomes. And you've all seen them. You've all seen pictures of it. You'll see a photo of, like, here's chromosome 1, here's chromosome 2, here's your X, here's your Y. They already had karyotypes of chimpanzee and of humans, and they say, look, you can line them up, and this one looks like this one, and this one looks like this one. But there's a huge, huge difference between human and chimp chromosome arrangement in that, well, the chimps actually have more.
Hey,
correct. Yeah, obviously, that's actually true. But the point would be that if there was a fusion event there, excuse me, when this happened, we would be able to see similar sequences that we would see with the other apes, and they're not similar at all.
Yeah, no one shares the same type.
No,
you were talking about the majority of people. You were talking about the majority of people over that one guy who looked into something. Now Joe asked in the side. Yeah, Joe asked in the side chat. In that particular instance, what does looking into something mean?
Well, they have software. Like you think about how many base pairs. When you talk about the human genome, how many sequences are in there? So they've for years now been studying they've got sequencing computers. So we have the human genome. We've heard this expression. We have the human genome mapped out. But then how do you read the map, right? I think that if somebody told you that the state where you live in is mapped out, and then you could look at it and say, oh yeah, well this dot is a city and this little squiggly line is a highway. But how does a person read the map of the genome? And it requires this specialized database software that you can go in. And then, well, so what are you going to see in the map? You're going to see A, T, T, C, G, T, A. Well, what does that mean? Again, Crew, please, the echo is just torturous. So then the question is then, how do you read that map? And of course, on the map, they have, when you look at the human genome sequence, the sequences in the human genome, there are things that encode for different things. So on on part of chromosome number 5, and again, I don't have it memorized, so I'm obviously going to get this wrong. But for example, the gene for eye color might be located in a particular place on chromosome 5. So this database software helps you go through and to look for these familiar gene sequences and know where these things are and kind of interpret these things. And of the regular features that they can recognize it with the software, telomere sequences, so those end cap sequences, are kind of a recognizable pattern, and centromere sequences. So when the chromosomes split apart, the centromere is kind of the central point that's get pulled in one direction when the cell goes through mitosis and splits, and then there's like a little thread that sort of hooks onto it. So that portion of the DNA is sort of functional or structural. Both the end caps, the telomeres, and the middle points, the centromeres, they're very functional, and they can't be there to do the things that DNA does, like coding for proteins or being regulatory genes. So when I say they've looked into it, these are people that really understand how the software works, and they can look through the sequence and say, oh, this is a telomere sequence, this is a protein coding sequence, this is a regulatory sequence, here's a centromere sequence. And so people who are experts in doing that have looked into it, and they say no, the gene he's talking about is there, but it's not across the telomere sequence. It's before the telomere sequence.
Okay, so what material can you show to back that up? Because there's nothing out there at all showing that the sequences are the same.
Yes, there is.
Okay, what can you show to back that up?
Do you know who Ken Miller is?
Yeah, I know what Ken Miller is. We showed a video of the guy debunking what he said. But I'm hearing a lot. Well, yes, we can do this, we can do that. But where's the evidence supporting it?
But if you read the actually, I've got a book for you if you want me to recommend it. It's called Finding Darwin's God. It's actually a really good book. It goes under, for people who may not be the most scientifically understandable, I make it understandable to them, into why there was a fusion event and why it causes what it caused. Now, the thing is that the reason that we know it's a fusion event and why it affects us is because the apes, the second chromosome, the DNA is so exactly matched apart from that if that didn't occur, evolution would be disproved. That's why it's important that we have mapped this stuff. And he goes into great detail of why it's important, what it proves, what studies they've done to back this up, because they've not just assumed it. They've studied it for many, many years.
Now we've mapped the genome, but that's not the point. These genes at the time, they were considered pseudogenes, right? We didn't know what these genes were actually used for at the time, and even now they're not found in any other chromosomes in any apes whatsoever. I mean, we don't find these sequences anywhere. So
which genes are you talking about?
I'm talking about the sequences of genes before the supposed fusion event and afterwards.
Okay, what about them?
What do you mean, what about them? If it fused right there, then that's where the location would be, where the two chromosomes fuse. So if you have two chromosomes right there at that part, you would be able to look at the sequences between both the chromosomes, right? You got one chromosome that's fused by the telomere, so both of the chromosomes are 64,000 sequences before that and after that. Those sequences are not found in any of the great apes. They're only found in humans. There's nothing indicating at all that this was a fusion event because there's nothing similar with any of the apes whatsoever. That's the point.
Oh, so you're saying that there's genes near the purported fusion site that are not seen in apes? Right? Or not,
yes.
Okay. Okay, so they'd be unique to humans. Okay. And then I just, in my head, I just hear, so what? Along with the echo that I hear in my head, I just hear, so what? But what is?
What do you mean, so what? If we share 48 if apes had 48 chromosomes and supposedly one of our chromosomes fuse, that's the reason why we have 46 chromosomes. There's nothing homologous there indicating that it was we had 48 chromosomes at one point.
Okay, but if the other genes,
indicating that,
if the other genes, like the thing is, you don't expect the chromosomes to be perfectly the same. Otherwise, they wouldn't be different species than us.
Let me screen share something for you guys. I don't think you guys are gonna get this. This isn't. I'm not gonna screen share the video again, but I'm gonna.
I do want to ask why you're doing that. What I do want to ask is, what do you see at the purported fusion site? Like aside from what's before it or what's after, what do you see at the purported fusion site?
What you see at the fusion site is actually, I got into this actually earlier. You see at the fusion site a transcription factor. That's a transcription binding factor. That's what you see at the binding site. Do you need me to elaborate or something?
No, I thought you were going to. I just want to ask you that real quick. I started eating my soup, my now cold soup, because I thought you were going to screen share something, so I muted.
Yeah, that's actually another point that we keep on overlooking as well, because what that part is, the very spot where you're finding the supposed fusion site, that's actually a very important part to where you're seeing it control the rate of the information in the DNA to RNA. So it's not like, I mean, it's not like some vestigial, whatever. It's actually a very important part Hold on a second. Can you guys see what I'm screen sharing?
Oh.
It's always my favorite moment. No, we can't.
Can you broadcast CRISPR?
Oh yeah, give me 1 second.
Hey, did you see the thing I just posted in the side chat?
You guys see this?
Yeah.
This is the human chromosome right here. A human chromosome fusion site right over here. And this is supposed to be the cryptic centromere. This right over here is what you see with the chimpanzee chromosome. Do you notice how how it looks completely different?
Yeah, it's like a totally different color.
No, I'm not asking you that. Do you notice the sequences to where they're different?
Yeah.
Well, one of them is a sequence of pink, and the other one is a sequence of kind of an orange.
Okay, well, you know that this is the fusion site right over here, right?
Right.
You know what I'm looking at on my screen? I'm looking at a cartoon stick drawing. So, are you sharing the right thing?
What you're looking at are the chromosomes within the chimpanzee, right? And you're looking at the human chromosome site, and you notice that the sequence right here is not the same thing.
Roddy, whereabouts did you get this from?
What are you screen sharing?
I'm showing you.
Looking at any
I'm screen sharing you the human chromosome 2 fusion site right over here, and the sequence that you would expect it to be, it's not found there.
It is.
Okay, you're saying that it's there. Show me where it is.
Well,
I can't show it that because I don't know where that
went away.
From.
Oh,
sorry.
It's gone.
Oh,
okay, you showed us a diagram, kind of a cartoonish.
That probably wasn't a good enough one.
Yeah. Sort of a cartoonish diagram of things showing the general order, and the order was what you would expect. I mean, again, it was obviously a very simplified diagram, but that was a simplified diagram of what was expected.
Why are you saying that? Why would you be saying that that's what you would expect?
Well,
what in there is what you would expect? What is in there? Why do you keep saying that? Like, what in there is what you would expect?
I'll put it up again, and let me look at it because I want to see the order that they're in. I mean, it doesn't show this is a telomere.
Hold on a second. I'm trying. There was another one that I had here on my documents, man, but I think I deleted it. So I'm trying to find the other one because this was a really good comparison. The picture that I had, it was comparing the ape chromosomes with the human chromosome 2 fusion site.
All I'm hearing is that what we're getting is that people who study DNA and chromosome organization, karyotypes, all this stuff, for many years now, have looked at these things and said that there are a lot of similarities between human chromosome 1 and, for example, chimpanzee chromosome 1. And when I say similarities, I mean that the genes appear in the same order, roughly speaking. There are some places where the order is completely reversed around for a small stretch, and then it picks up, and that echo, honestly. Okay, so and then you see, like, you can line up the chromosomes and say, well, this one's the same as that one, this one's the same as that one, but there's this one really huge difference, and that's humans have this chromosome 2, which is very long, and the chimpanzees have 2 other chromosomes that their length is just slightly longer when added to human chromosome 2. And a lot of the genes across those 2 separate chromosomes match human chromosome 2. But of course, there's a lot of differences. We're different species, and we have a long history apart from chimpanzees. Then, upon sequencing, they find telomere and a centromere and a telomere, centromere, telomere, and they come to the conclusion, hey, this sure looks like a chromosome fusion. And what I'm hearing in response from you guys is, well, there's no proof of that. Well, no, it's not really a telomere.
That's not true at all.
No,
you're operating on a trauma. That's not what's true at all.
Okay, well,
I made it very, very clear that that binding site, that fusion site, where it happened with the telomere-telomere fusion, doesn't happen anywhere. that whenever it happens, it's dangerous. So we have no reason to believe that this was actually a fusion event. Every fusion event that we see, every single one of them, to where they actually happen in thriving living organisms, are either a satellite DNA to satellite DNA fusion event or a satellite DNA to a telomere fusion event. There is no telomere to telomere fusion event, and we've asked repeatedly. Hold on a second, Hogtie. Hold on a second. Every single event that we have seen has been dangerous and triggers apoptosis whenever that ends up happening. The cell would self-destruct if that were actually to happen. That's where the evidence is being provided right here. We know that. So if you're going to sit over here and say that this was actually a fusion event, you got to have evidence for that rather than saying, well, I see a pattern here that I'm going to use my imagination for.
You saying, no, it's not your imagination, it's your vision. It's sequencing computers.
Hold on, I'm not finished talking.
You interrupted me, and then you've said the same thing over and over again. Okay, then keep talking.
I'm not finished talking.
Next time it's my turn.
I'm not finished talking. There was a lot more problems with what I brought up before. The transcription binding site that you find located right there, it's an important factor that tells us why you're seeing that set right there to begin with. I mean, there's a lot of other things that were talked about here that you're missing and overlooking. Okay, we're not concerned with, you can look at patterns and say, well, therefore evolution was behind it. We're concerned with knowing if this type of fusion events can actually happen.
Okay, but the thing is, you're looking at one. You're looking at one, and you're saying,
no, we're not.
Okay.
No, we're not. We're not looking at a fusion event.
Seriously.
We are not looking at a fusion event, and I explained why. Do you remember what I said earlier about why it's not a fusion event?
You said there is no evidence it's a fusion event. There is no evidence it's a fusion event. No evidence it's a fusion event. Right after I've said, look, people look at this, they see telomere, centromere, telomere, telomere, centromere, telomere. You might disagree with the conclusion, but you cannot make the claim that that's no evidence.
You don't see two centromeres.
Okay, what was the thing about interrupting people? What was your policy about interrupting people?
What was your policy about
interrupting people?
No. Can I do it?
Okay, so this is why I said, what's the point? I mean, CRISPR has a lot of useful information about this. He's more of a chromosome guy than I am. You get to this point, Ronnie, where you just, when you know that the other person's saying something that is going to be something you can't address, you just start interrupting.
All right, let's hear you. The problem is that you're strawmanning us. I see how you're speaking to my audience.
You strawman me.
You know that we're not. No, look, you keep on wording things in a way so you're saying, well, only one scientist says this. The vast majority have looked at this and disagree. That is an ad populum fallacy.
No, it's not an ad populum fallacy.
Hold on. It's not. Keep on wording things in a way to where you're strawmanning us inside of the video.
It's based on strawmanning us.
Hold on a second.
Looking at the sequences. Why do I have to hold on a second when you don't? When you can interrupt me after 2 sentences?
Because, well, you were interrupting me beforehand, so it's my hangout if you don't like it. Okay. There's one centromere found in there. You haven't provided any evidence. There are 2 centromeres found. Centromere, telomere. You haven't provided any evidence for that. You rush past really quick and make that statement, but you don't justify the statements that you're making. So if there's 2 centromeres, point out two centromeres, because remember, that's what you said. You didn't stay here that there was a vestigial centromere. You said that there was 2 centromeres, centromere, or telomere. So let's hear what you got to say.
What I said was there are 2 centromeric sequences. And then later you asked me, you said something like, well, there should be 2 chromosomes, and that totally confused me. And then, you meant centromere. You said, yes, sorry, I meant centromere. And then I said, well, no, of course there aren't 2 functioning centromeres. There's 1 functional centromere and 1 what you might call vestigial centromere. I think they actually have a different word for it. But so what I said was, I didn't say there were 2 centromeres. Okay, sorry. When speaking offhand and I go telomere, centromere, telomere, telomere, centromere, telomere, that's just, I mean, that phrase is already ugly long. I suppose I should say telomeric sequence, some stuff, centromeric sequence, some stuff, telomeric sequence, it's just blah, blah, blah. Okay? It's too long to say it that way. So when I say centromere, I don't mean there's 2 functional centromeres. What I mean is, and I've been clear about this, there's centromeric sequences in there. It's very clear that this is like a deprecated centromere.
No, there are degenerate sequences. There are degenerate sequences that you're dubbing is a vestigial centromere that was there. That's what's happening. We don't know if it was actually we don't know if it was actually a centromere. If you're going to say that it was a centromere,
we don't know if the world was created 5 minutes ago. Ortholog in a chip.
Yeah.
I couldn't hear anything you said, Isaiah.
Can you hear me now? Am I coming through clear?
I think we should give CRISPR a shot.
Yes, please, if I can.
Whoever, I don't really care.
Okay, so if possible, I'm going to try to approach this from a slightly different direction. So previously, the chimpanzee chromosomes had been annotated as chromosome 12 and 13, and they're now annotated as chromosome 2A and chromosome 2B. Okay, you can agree with that, right? That you agree that that's the way they're currently annotated?
Yeah, that they're
okay. Okay, so my question is, would you agree that the majority of the genes that are found on chimpanzee chromosome 2A, as it's currently annotated?
I'm gonna be listening, but I gotta go answer my door really quick, all right? So go ahead, keep talking.
Sure. Are homologous with the genes that are found on human chromosome 2P. That is on the one side of human chromosome 2. And that the same is true regarding chimpanzee chromosome 2B, homologous to human chromosome 2Q, that is on the other side of human chromosome 2. That those genes they match each other between the two chromosomes in chimpanzees and the one chromosome in humans. So
not talking to see. Christopher, do you happen to have a diagram, or I want to say this out loud, sorry for interrupting you, but because that was the point I was trying to make before about how that the genes match up, but I don't think it's really helpful to just say that. I wonder if you or somebody who's watching can send a link to a diagram that shows that, like, here's a chromosome, here's this gene, that gene, this gene, that gene, with regard to 2P and 2Q in humans and 2A and 2B. Oh. By the time sorry. I was asking the question. He put a link in the side chat, which you answered to. Thank you.
Yeah, so that's originally from Yunus and Prakash, which was published in 1982. So.
Okay, so you're showing me the. What are you showing me right here? The.
Yeah, and see those bands with the banding colors. What that represents is different regions of is it from, I think, from fish, I think. So this is not they hadn't actually sequenced these at that time, right? They just done fluorescent in situ hybridization experiments, if I remember correctly, that is, and that's how they developed these banding. Somebody else who's more informed on the genomics can correct me on that, but anyway, yeah. So that is the homology, not the sequence homology. It's a sort of super sequence homology, if you will, a broader structure.
Well, somebody just came to the door, and I'm actually going to be going on a trip in a little bit to Texas. So I'm going to have to cut this hangout short in about a half an hour because I'm going to be taking off. But I just want to say that the binding site that you guys are referring to, again, that's not necessarily what you guys are looking at. You see a transcription factor where you have to deal with the sequence of, or the sequence rate or the control rates of the information to DNA to RNA. So that binding site that does not necessarily show a fusion event. And even if we're going to look at the vestigial, of what we're going to claim to be a vestigial centriole, I think we're going to have to need some evidence supporting that. We've been on this. We've been on this for a while over this issue of the chromosome 2 fusion. We also have to start talking about the other part of this section of this hangout, which is intelligent design. So do you guys want to go in that direction?
You're literally going to cut him off right when he's giving you the evidence you asked for.
Right.
I'm actually screen sharing an image that shows the banding, not necessarily the sequences, but the banding.
Yeah, and so.
Okay, so how is this evidence now for chromosome 2 fusion?
So what I want to do is just momentarily, we've been talking about the vestigial telomeric sequences, centromeric sequences, and all that, but I want to step back from that for a second. Would you agree that the genes that can be found on chimpanzee chromosome 2B are homologous to the genes that are found on human chromosome 2Q? That is one side of human chromosome 2.
I would agree that you might find a few sequences that
almost all of them
might appear similar,
but there's 64,000. I'm concerned with the 64,000 base pairs or base sequences.
I'm talking about
wow.
It's
I'm
614.
talking about 100
Do you
million
see
base
it?
pairs.
That's it. That's exactly it right there.
I'm talking about 100 million base pairs.
100 million base pairs versus 64,000, and you want to. This is exactly.
Okay. If you're shut up, Hogtie, come on.
No. This is a light bulb moment. This is
great. It's a light bulb moment. Can I hear him make the light bulb moment? Because I don't want to hear you interrupt. Okay?
No, because he's telling you about something, and you want to talk about.
Okay, I think I can manage this on my own.
This is getting ridiculous.
Come on.
Listen. Stop it for a second. I'm going to turn down your volume because we're going to make this perfectly clear right now, right? Every time I try to start talking, you're bursting something out. You're bursting crap out like, well, we just showed the evidence, and you're rushing past certain statements, and you're not justifying anything that you're saying. And you're actually contradicting yourself, like what you said about the centrioles earlier. The centrioles that I was talking about earlier, you do not find the telomere sequence of the centrioles. That's not what you're observing at all. You see a degenerate form that might appear to have been a centriole, but it's still degenerate. I mean, how are we going to say that it was actually a centriole anyways? But anyways, I would love to see if they're sharing the homologous sequences. So go ahead and go at it.
Yeah, so what you just said is fine. Okay, so what I want to do is I want to ignore the question of the vestigial sequences for a moment. So I want to ignore the question of whether or not we can see the telomeric in the middle or the second centromeric sequence. I just want to ignore that for a second. And so what I want to focus on is the fact that if you look at the one side of human chromosome 2, what you see is all of the genes, okay, all of the genes that are on chimpanzee chromosome 2A. And if you look at the other side of human chromosome 2, you see all of the genes that are on chimpanzee chromosome 2B, right? And in the same order for the most part. And so...
No, I disagree with that 100%.
Okay.
You might find certain sequences that are similar, but that wouldn't surprise me at all.
I'm sorry. I mean, I can show you this. That it's just
okay.
All the way down.
I'd like to take a look at it. Is there like is it like going to be some sort of peer-reviewed article that I can take a look at?
Yeah, there's quite a few. I don't have them pulled up right now, but I'm certainly send them to you.
Right, because none of the sequences that you're finding before and after this chromosome, before this supposed fusion site.
Oh, I'm sorry.
Those sequences, they're not found anywhere at any group at all.
I'm sorry. Understand?
Yes, they are. I have a paper on that.
I say it, please. I understand, True, that I'm not talking specifically about that fusion site. I'm talking about the entirety of the one side of the chromosome and the entirety of the other side of the chromosome. I'm ignoring the fusion site for the moment.
I know. I'm hearing your claim, but I need evidence to support that claim. That's what I'm asking for. I mean, do you have any piece of paper that tells me that all of the base pairs, all of the sequences, they are homologous to the human chromosome? Do you have anything supporting that? Or the chimpanzee chromosome?
Yeah, I do, but you're gonna have to give me a few minutes to pull it up.
All right. Well, then I don't have it. I see, Hogtie. If you would have calmed down, we could have worked through. You see. Okay. So, I mean, do you guys? I mean, I know I'm ignoring everybody else here. Antonio, you haven't really gotten to speak. David, you haven't been able to talk. You guys have been here longer than everybody else. You guys want to move on to the situation with intelligent design? Or do you guys want to stay on this issue? You guys hear me? Hello?
You speaking to me?
Yeah, David. Did you want to stay on this point, or you want to move on to intelligent design?
Well, not really. I'll just, I mean, it just seems to me like most of the evidence is just based on assumption from either side, really.
Did you see the link I put?
Hold on.
So it's never really going to be a thing where you can...
I couldn't hear you at all, David. Well, anyways, guys, Veckel just came in. So, well, I'm gonna try to end the Hangouts, guys, because my phone's ringing off the hook, and I got to get ready to back up here. So, who hasn't gotten to speak yet? Veckel just came in. Veckel, you want to say something?
Yeah, just chiming in, Mr. Taylor. I just walked in through the door, and don't really know what the conversation's about, but you know, just I like listening.
Mm-hmm.
And yeah, sorry, I don't have anything to add right now.
Okay, well, I want to make it clear, none of the criticisms, and I know everybody's going to disagree with them, none of the criticisms were dealt with that I would like to have dealt with. None of them were. I asked about the telomere fusion, if there could be other examples given, because most examples, they're dangerous or hazardous to life. I don't disagree that there are fusion events happening, but they don't happen with the telomere fusion, and if they do, they're dangerous. They're usually satellite DNA to satellite DNA fusion event, or they're a satellite DNA to telomere fusion event. We don't have any examples of that. The example that Hogtie basically gave was that, well, just because we haven't seen this happen, that doesn't necessarily mean that it couldn't happen, basically using his analogy with somebody that might have gotten shot in the heart. That doesn't really answer the question. You can't just assume something that hasn't had data yet supporting it to be the case. Again, I disagree with this actually being a fusion event anyways to begin with. I think that fusion event is actually the start of a very important process that happens with cells, or with control rates of RNA and DNA. That's a very important part right there. He gave an example of there being the telomere and the sequence with the centrioles. We do not see two centrioles here. That's what's really important here. We find a degenerate types of sequences to where we're superimposing. Well, therefore, this was the remaining parts, I guess, or the degenerate parts of what used to be another centriole, but we have no reason to believe that except for presuppositions. So I can come in here assuming that this used to be a centriole because I can imagine it to be so, but that doesn't necessarily mean that it was so. And usually in the future, these types of speculations or superimposing on information ends up being confirmed to be incorrect. So, I mean, I haven't seen any example of these types of fusion events actually happen that are actually safe for living organisms. I haven't seen any examples of there being, because I wouldn't disagree with this actually. I wouldn't disagree that there were actually certain sequences that you could find that would be very similar to chimpanzee chromosomes or sequences, but that's definitely not going to be consistent all the way across the board. I would disagree with that. There might be certain parts of, that you would find scattered across a chromosome, but all of the sequences are not going to be the same thing. So I have no reason to believe that this was a chromosome fusion. I think that the case has been made solid and strong here if people use a little bit of wisdom when they're watching. God bless everybody. Take care.